I'm Carter, and I'm a project officer for the Freshwater Habitats Trust, and I'm going to talk to you today about the results of our eDNA surveys as part of our citizen science testing the water project. A little bit about Freshwater Habitats Trust. We started life back in the late 1980s as Pond Action, and subsequently we became Pond Conservation. And then in 2013, we actually changed our name to the Freshwater Habitats Trust because we are concerned with all freshwaters, including those that are small and undervalued. So we didn't want to just focus on ponds. We also wanted to look at streams, rivers and ditches. Now, we are an evidence-based conservation charity with a very strong science grounding. We are highly strategic, so we target our work where evidence suggests it will be most effective. And we work in partnership with people, communities and organisations like the Brecks Edge and Rivers Landscape Partnership to get the best results for freshwater wildlife. Fen Edge and Rivers Landscape Partnership Scheme is a new landscape partnership scheme to conserve and celebrate the unique heritage of the Norfolk and Suffolk Brecks. Over a 5-year delivery period between 2020 and 2024, working with partners and communities, the Brecks Fen Edge and Rivers Landscape Partnership aims to deliver a minimum of 24 projects. And we are leading on one of these projects, Citizen Science Testing the Water. So the aim of testing the water is to get new information about the project area through surveys and to involve people in the collection of important biological and environmental data needed to protect life in freshwater. And we've been doing this by carrying out citizen science surveys using cutting-edge eDNA kits to test for vertebrate species in the Brecks water bodies. And quick-use clean water kit is to test for nitrate and phosphate pollution in the water bodies. It will help to achieve 3 broad aims. To map the extent of clean, unpolluted water, providing a baseline for long-term monitoring. To increase people's engagement with and understanding of the exceptional freshwater heritage of the Brecks. And to inform the delivery of river restoration activities and guide future landscape conservation activity in the scheme area. BFER landscape scheme area. So this borders on the counties of Cambridgeshire, Norfolk and Suffolk, and includes the river catchments of the River Wizzy up in the north, the River Lark down in the south, and in the middle the Little Ouse. eDNA survey for aquatic wildlife. Well, eDNA is a powerful tool for surveying a range of aquatic creatures without the need to actually catch the animals themselves. It has been shown to be effective in a wide variety of aquatic ecosystems, so ponds, lakes, streams, rivers, estuaries and even oceans. And it can be used either to detect the presence of a particular species or to survey communities of organisms. Surveys, we use a multi-species kits from Naturemetrics, and these were set to detect vertebrate species in the water bodies, so that includes mammals, fish, amphibians and birds. The site selection, the monitoring network, we selected 71 sites spread out across the Brecks area, and this included all the different water body types, so the ponds, rivers, ditches, streams, lakes and cut-off channels. In April 2022, we ran an online training course to train volunteers up to go out and sample these water bodies. So once trained up, we posted out an eDNA kit, you can see here, and some clean water kits, as well as a very simple sort of site pack so that they could find the water body they wanted them to sample, and it's got any landowner information on as well. And these surveys were carried out by volunteers between April and the end of June 2022. The results. The 71 survey sites, we actually got 57 survey results back. So quite a few of the original surveys came back as void, and this is for several reasons. It could have been that there was not enough water filtered through the sample, so not enough DNA captured, or it might have just been non-target DNA that was captured, such as human and domestic pets. But you can see here the array of different water body types. So you can see the different colours. So streams, rivers, ponds, lakes, ditches, and cut-off channels across the Brecks area. The volunteers, when they went out to take photographs of them actually doing the surveys, which is absolutely brilliant to see, and a lot of them posted on social media as well. So that was absolutely great. Photographs, for those of you who are not familiar with the eDNA survey, so basically the volunteers collected water samples in a large plastic bag and then filtered the sample using a large syringe through a filter to capture any DNA from the water body on the actual filter itself. The preservative was then put in the filter and capped off and then sent back to the lab for analysis. Looking at the vertebrate species across all the water bodies types, so 68 species in total were discovered in this survey. And out of this, the percentage of vertebrate taxa found across all the water body survey sites, as you can see in this pie chart, 38% were fish, 38% birds, 16% mammals, and 7% amphibians. And if you look at the bar chart below, this shows the percentage of taxon groups found in all the water bodies. So for the fish, 98% of the sites contained fish, 88% fish, 98 birds, 46% mammals, and 51% of the survey sites found amphibians, and that's out of 57 sites. And then you can see on the map on the left, this shows the species density. So this ranged from just one species found to up to 27. So the maximum amount of species found in any of the survey sites was 27. Look at the amphibian species. Well, we actually detected 5 amphibian species in total. And this included the common toad, the common frog, the smooth newt, the great crested newt, and the pool-stroke marsh frog, with the common toad the most abundant, found in 19 of the survey samples, followed by common frog at 16, then smooth newt at 9, great crested at 3, and the pool-stroke marsh frog at 2 sites. The only newt species we didn't detect was a palmate newt, which is not unsurprising as the species is generally not recorded in the Brecks area. It's the wrong sort of habitat. So just a little bit about the pool-stroke marsh frog. So these species are part of a group of frogs called the green frogs, and they are so closely related that the DNA markers couldn't detect them, the species apart. Now, the marsh frog, that is a non-native species, and as far as we're aware, there's no records in the Brecks area for this. The pool frog is interesting because this is actually a native species, and this was only discovered a few years ago at a time when the animal went extinct from the UK. And we discovered it was a native species from fossil records. So, and since then, pool frogs have been reintroduced on one of the sites where records suggested they previously existed on Thompson Common in the Brecks area. The common toad was found in all water body types, both standing and running waters, although their preference is for standing water bodies, so sort of gravel pitches and larger water bodies. They're actually toxic or distasteful to a lot of predators, including fish, so they can actually survive quite well in water bodies that have quite a few predators. The common frog was also found in all the water body types, and if you look at the map on the right hand side, you can see that there's a lot, the sort of turquoisey green, that was where the frog and toad were both found. But they're both, if you look at all the greeny blue squares, they're fairly well abundantly spaced across the Brecks area. The smooth newt was found in 67% of standing water and 33% of running water. And the great crested newt was found only in standing water. And you can see again the smooth newts reasonably well distributed if you look at the little brown boxes, whereas the great crested newt, they were only found in the sort of wetland rich semi-natural habitats on the Stanta military training area and Thompson Common, and not in the more widespread agricultural landscape. And the little pool frog, as we've discussed, they were only found in standing water as well. So again, that's interesting because one of the samples was found in a pond close to Thompson Common, so we could pretty well assume that that was a pool frog. But one of the other sites, if you can see the little black spot for the pool frog, were found on a site just near Thetford. Now that is a site that's never been recorded there, and the site managers have never discovered them. It's a very well monitored site, so the jury's out as to whether that was a false positive or not. It's very highly unlikely that they've found their way from Thompson Common, but there is an outside chance that they may, if they are there, then they have been introduced by a member of the general public, because both pool frog and marsh frog can actually be bought as pets, and people have been known to release these creatures, similar to the terrapins, into ponds or the water bodies in the wider landscape. 26 fish were detected in total, so quite a lot, and the most abundant were the 9-spined and 3-spined stickleback. But the creatures of interest here, we've got the European eel, which is a protected species, and these are migratory species, and they migrate from the Sargasso Sea to inland water bodies in the UK to breed. And the lamprey species, similar to the marsh stroke pool frog, these were identified as brook stroke river lamprey. And again, these two species are so similar, closely related, the eDNA markers couldn't tell them apart. So we're not 100% which ones they are. However, the river lamprey, like the eel, these are migratory species, whereas the brook lamprey is not. So they spend all their time in water bodies in the UK. The European eel was found in the rivers the Lark, the Wizzy, the Little Ouse and the Tet, as well as the lamprey species were also found in these rivers. And as you can see from the map, if you look at the blue spots, so out of, I think, the European eel, we found 9. Were found in 9 sites, and the lamprey in 11, and out of those, 8 of those, both species were in the same water body, so obviously have a preference for similar type of habitat. But they were also, interestingly, found in standing water bodies as well, so not just running water. And the sticklebacks were found in all water body types, which is not surprising because these little fish are very widespread in the UK and are found generally in all water body types. Out of all the mammals, 10 were identified, and 3 of these were identified as aquatic mammals. So we have the water vole, the otter, and the water shrew. And the most abundant out of these was the water vole with 13 sites, the water shrew at 3, and the otter at 1. And as well as these aquatic mammals, and a variety of non-aquatic mammals were also discovered in the eDNA. So we had red deer and muntjac deer. We had wood-stroke yellow-necked mouse, the brown rat, red fox, mole, and grey squirrel. were also found. And these animals were most likely using the water bodies to drink and wash. Distribution: the water vole was found in all water body types, including the rivers Lark, Little Ouse and Gadder. Whereas the otter, we just found one otter, and that was found in a pond adjacent to the Little Ouse, which is also connected to the river. And that's quite interesting because the water vole was also found at that same site. So if you see on the map at Brandon, you can see there's a little yellow and green square overlaid. So that's where the otter and the water vole were found. And the water vole was also found upstream as well. But just because we only detected one otter, that's not to say they're not more widespread. We did have a couple of records of mustelid species, and they could very well have been otter as well, but we can't say for definite. And the water shrew, they were found at 3 sites in the River Gadder and 2 streams. And interestingly, if you look at the map, if you look at the pink square, all the water shrews were found in the same water bodies as the water vole. 26 species altogether. The most abundant were the waterfowl species, and again, unfortunately, the eDNA markers were not strong enough to detect which actual species these were, but the group does include swans, ducks and geese, which are the most abundant waterfowl species to be found in the UK, and are found in most water bodies, including those on the Brecks. So that's not surprising. So 47 out of the 57 sites detected waterfowl species. We also detected 5 aquatic bird species, so the moorhen, the grey heron, the common coot, cormorant and water rail, as well as an abundance of non-aquatic species, as you can see from the chart here. Distribution again, the moorhen was found in all water body types and is pretty well distributed across the Brecks area. And this isn't surprising because this little bird can be found in nearly all small water bodies. You've got a pond, there's a good chance that you've got a breeding moorhen on there. The grey heron was found in ponds, lakes, rivers and streams, and the coot was found in ponds, lakes and rivers. And interestingly, although a cousin of the moorhen, the coot tends to prefer larger water bodies. And the actual rivers they were found on, they detected on, were actually adjacent to quite large sort of lake sort of sites. So that's quite interesting. And the cormorant was found in lakes and rivers, and the water rail just at 2 sites in rivers and ditches. Look at water quality. So we asked the volunteers, as well as taking the eDNA sample, to take a water sample at the same time. And you can see from this map, the little red dots are indicating polluted water, whereas the blue dots are indicating unpolluted clean water across the Brecks area. Split that out between standing and running water. You can see here on these maps 71% of the standing water had clean water and only 17% of the running water had clean water, as you can see on these maps. Versus running water, as we've seen, out of the vertebrate taxa, 38% fish, 38% birds, 16% mammals and 7% amphibians. And you look at this map, and this shows a percentage of species using standing and running waters. So you can see here at the top, out of the 7% amphibians, 7%, so all the amphibians were using standing water, but they were also, 4% were also using running water. Now with the mammals and the birds, again, they're both equal, so 16% and 38% equally using running and standing water. Whereas the fish, 38% were using running water and 31% were using standing water. So across all the species in total, 97% were using running water and 93% were using standing water, and that's out of 68 species in total. The results of our eDNA surveys reveal that a large number of vertebrate species are utilising the freshwater environment in the Brecks area, and as well as the fully aquatic animals such as fish and aquatic and semi-aquatic animals from the mammal, bird and amphibian families, an abundance of non-aquatic species were also found to be using water, most probably to drink and wash, which highlights the importance of freshwater habitats in the environment. The results also reveal that most of the species detected were found across all the water body habitat types, including standing waters, so ponds and lakes, and running water, so rivers, streams, ditches and cut-off channels. This suggests that the whole freshwater environment is an important resource for all vertebrate species. And some species, toads for example, have a habitat preference for standing waters, but in this study they were also found to be utilising running waters. Now, most freshwater plants and animals have evolved over millions of years in a world where the natural level of nutrients in ponds, lakes, streams and rivers was very low. And when more nutrients like phosphate and nitrate are added to the freshwater environment, this can cause profound changes and the freshwater wildlife can struggle to adapt. It is actually very difficult to improve the water quality of running waters as rivers and streams drain large areas of land and are exposed to multiple sources of pollution from both urban and agricultural areas. Now, ponds and lakes are also affected by the surrounding land use, but they naturally drain smaller areas of land, so are less likely to be affected by pollution problems. So building up the network of standing water bodies in the landscape by creating more ponds in clean catchments could therefore offer up a solution to increasing freshwater biodiversity in the Brecks area. And as the study has demonstrated, most of the species detected were found across all the freshwater habitat types. Even species traditionally associated with rivers, such as the European eel and the lamprey species, were detected in standing water bodies. So creating more clean water ponds in the landscape could therefore provide a refuge for a multitude of species away from the pollution issues associated with running waters. Clean water is vital for freshwater biodiversity, and these potentially unpolluted sites could support rich and valuable wildlife communities. I'm going to finish off with sharing a few photographs taken by our wonderful volunteers. So we can see here the different ponds, streams, rivers and ditches. Here we have a few more from our wonderful volunteers. And thank you for listening.